Hello students, welcome to this session. Let's study cell part 2 biomolecules. Biomolecules, one is proteins. They are defined as polymers of amino acids and contain carbon, hydrogen, oxygen and nitrogen. The classification simple proteins, then conjugated protein in which simple plus non-protein substance that is prosthetic group is present. So examples are phosphoprotein, lipoprotein, glycoprotein, nucleoprotein, example DNA, RNA, porphyroprotein, example hemoglobin. Complete protein contains all essential amino acids which require to maintain normal life and growth. Partially complete proteins can maintain life but not growth. And incomplete proteins lack one or more essential amino acids, cannot maintain life. The functions of proteins: protein nutrition, osmotic balance, water balance, buffering action, transport of lipids, blood coagulation by hydrogen, transfer of oxygen to tissues by hemoglobin, transfer of carbon dioxide to tissue from tissues by hemoglobin. Importance: the proteins are building blocks of the body. They are essential for growth and repair of the body. A major constituents of bone, tooth, skin, nails, hair, blood cells. Proteins serve as a source of energy. DNA and RNA are proteins presenting nucleus and cytoplasm. The structure of proteins is of four types. That is primary structure. The number and order of amino acids in polypeptide chain is referred as primary. So dipeptide 2 amino acid, tripeptide 3 amino acid, tetrapeptide 4 amino acid, polypeptide more than 10 amino acids. Heme peptides. Then the secondary structure, the folding of polypeptide chain into a specific coil structure. The tertiary structure is twisted chains of protein into specific layers, and quaternary, some proteins will display fourth level of organization. Here, several primary, secondary, tertiary structure are combined from the quaternary structure. The properties: denaturation, chromatography, and electrophoresis. So denaturation is when protein undergoes changes in structure of composition like heat, mineral, acid, alkali, shaking, grinding, alcohol, ultraviolet rays, ultrasonic rays, waves. Then chromatography and electrophoresis, that is separation of the proteins. Digestion and absorption. So in stomach, because of the pepsin in gastric juice, and hydrochloric acid breaks down proteins into polypeptide. Then pancreas, trypsin breaks polypeptides into peptose and propeptones, and then into amino acids. And small intestine, leptin helps to break polypeptide into amino acids, and then amino acids are absorbed into the blood.
So today we will talk about proteins. So proteins are polymers of amino acids and they contain carbon, hydrogen, oxygen and nitrogen. So they are also called as polypeptides. So amino acids combine with a peptide bond and many peptides form the polypeptides. So actually they are of 3-4 structures like primary, secondary, tertiary and quaternary. So polypeptide chains are there and all these chains together form the quaternary structure, that is proteins. Classification: it is simple protein, derived protein and conjugated protein. So simple protein means it is only protein. So examples: albumin, globulin or keratin which is present in nail, or collagen which is present in skin and bone, or elastin which is present in ligaments. Derived proteins, they are breakdown products of the proteins in while digestion. The conjugated proteins, they are combination of simple protein plus non-protein substance, and that non-protein substance is called as prosthetic group. So a few examples like phosphoprotein, it is a combination of protein plus phosphorus. Example milk, so in that casein is there, or egg yolk, in that vitelline is there. Lipoprotein is a combination of protein plus lipid. Example egg yolk. Glycoprotein is a combination of protein plus sugar. Example mucin. Nucleoprotein, protein plus nucleic acid. So example DNA, RNA. Porphyrinoprotein is a combination of protein plus metal, that is porphyrin group and metal. So example is hemoglobin, in which iron is there as a prosthetic group. Classification of proteins based on amino acids can be complete, partially complete and incomplete. So complete proteins when it contains all essential amino acids to maintain normal life and growth. Partially complete when few amino acids are missing, and then it maintains life but not the growth. And incomplete proteins means essential amino acids are lacking and which do not maintain life and growth. So they are incomplete proteins. Functions: they are nutrition, provide then transport of lipids, osmotic water balance, buffer action, and blood coagulation takes place because of the fibrinogen, and it prevents blood loss. Then respiratory function by hemoglobin, that is transfer of oxygen to tissues and transfer of carbon dioxide from tissues to lungs, sorry, from tissues to outside. Digestion takes place in stomach. So in that, gastric juice is there which contains pepsin and hydrochloric acid. So this pepsin breaks down proteins into polypeptide plus peptide. Then pancreas contain pancreatic juice which contains trypsin, and it breaks down polypeptides or peptodes into amino acids. Small intestine contains intestinal juice which contains erepsin, and that breaks down polypeptides or remaining part of the peptones into amino acids. And finally, simple form, that is amino acid, is formed from the proteins, and then that is absorbed into the blood. Importance: they are building blocks of the body. They are essential for growth and wear and tear of the body. They are present in bones, tooth, skin, nails, hair, blood cells. They are source of energy, and also they are present in DNA and RNA, which is present in nucleus and cytoplasm. Thank you.
Proteins are made up of units are called as amino acids. The classification: essential, semi-essential, and non-essential. So essential amino acids are those amino acids which cannot be synthesized in body at a sufficient level to meet body need. They must be taken in diet. Example: methionine, threonine, tryptophan, valine, isoleucine. Semi-essential amino acids are those amino acids which cannot be synthesized from other amino acids. Example: cysteine, tyrosine. Non-essential amino acids are those amino acids which can be synthesized in body. They need not be taken in diet. Example: glycine, alanine, cysteine, aspartic acid, glutamic acid, proline, hydroxyproline. The importance: amino acids must be supplied in diet to maintain normal growth and nitrogen balance and health. Aspartic acid, glutamic acid are involved in transmission of impulses in nervous system. Many hormones are amino acids. Example: adrenaline, thyroxine, T3. Glycine is required for synthesis of heme. Amino acids are required for synthesis of nucleotides. Abnormalities in transport of amino acids in cell results in disease.
Hello students, today we will see now our macronutrients, that is nutrients required in large quantity. They are: first is carbohydrates. So carbohydrates definition: they are hydrates of the carbon, hydrogen and oxygen group. So sources are like plants, leaves and all, and fruits plus wheat, rice, potato. So classification of carbohydrates is of mainly three types, that is monosaccharide, disaccharide and polysaccharide. So monosaccharide means they are simple sugars. One sugar molecule is there. Example is glucose, fructose. Disaccharides, they contain two sugar molecules. Example is sucrose, lactose, maltose. So lactose present in milk and sucrose present in sugar and maltose. Third type is polysaccharides. So they are more than 10 molecules of monosaccharides, that is simple sugars, present in polysaccharide. So they are of two types, homopolysaccharide and heteropolysaccharide. So if the sugar molecules are same, that is monosaccharide, they are homopolysaccharide, and examples are starch, cellulose, glycogen, dextrin. And if the monosaccharides are of different types, then they are called as heteropolysaccharide, and example is mucopolysaccharide. Now digestion of carbohydrates, usually it takes place in digestive system. So digestion of carbohydrates starts from the chewing mechanism, that is from the salivary gland which secretes saliva, which contains salivary amylase, and then that breaks down cooked starch into the maltose, that is disaccharide. Then in pancreas, pancreatic juice is present, and which contains pancreatic amylase, and which breaks down again the remaining part of the starch into maltose, that is polysaccharide is converted into disaccharide. Then in small intestine, the intestinal juice is secreted, and then there are two enzymes, invertase and maltase. So invertase breaks down sucrose, that is disaccharide, into monosaccharide, that is glucose, and maltase breaks down maltose into glucose, and then finally this monosaccharide, that is glucose, simple sugar is formed, and which is absorbed into the blood. Now we will see importance of carbohydrates. So carbohydrates are easily available and least expensive source of energy for the body. 1 gram of carbohydrate on oxidation gives 4 kilocalorie of the energy, and glucose is a main source of the energy. Glucose is converted into glycogen and stored in liver and muscles. And when the fasting state or when we do not take food, that time the glycogen is again converted into glucose and then maintain the blood sugar level. Muscles glycogen supply energy to muscles, and some part of glucose is converted to fat for energy reserve, that is in fats. And then because of this the roughage diet of the polysaccharides, there is no constipation and there is no gastrointestinal disorders. To maintain the bowel movements, this rough fiber food is important for the digestion. Thank you.
Carbohydrates are polymers of monosaccharides or simple sugars. They are the main energy source of plants and animal cells. Classification: monosaccharide, disaccharide, polysaccharide. So monosaccharide, they are simple sugars, single glucose molecule, that is glucose fructose. Disaccharide, two simple sugars. Example: sucrose, lactose, maltose. Polysaccharide contain more than 10 molecules of monosaccharides. Source of carbohydrates: plants, leaves, stem, wood, fruits, seeds, wheat, rice, potato. Importance: carbohydrates of least expensive source of energy. 1 gram of carbohydrate gives 4 kilocalorie of energy. Glucose is main source of energy. Glucose is converted into glycogen and stored in liver and muscles. Liver glycogen can be converted to glucose to maintain blood sugar level. Muscle glycogen supply energy to muscles. Glucose is converted to fats for energy reserve. Roughage constitute polysaccharides to reduce gastrointestinal disorder and to make bowel movements easy.
Hello students, today we will talk about lipids, that is fats. So they are organic compounds of combination of fatty acid plus glycerol. So classification is simple lipid, compound lipid and derived lipid. So simple lipid, they are esters of fatty acid plus glycerol. Example is fats, which is present in tripalmitin, coconut fat, animal fat, or wax, which is vitamin D esters or true wax. Then derived lipids, they are found in breakdown of the simple or compound lipids. So second type is compound or conjugated lipids. So they are esters of fatty acid plus glycerol plus other compound. So like phosphoric acid is present, then it is called as phospholipid. So they are present in nervous tissue, brain, liver or kidney. An example, they are cephalin, cardiolipin, plasmalogen. So they form a structure like mitochondria, or cephalin helps in coagulation, or they act as a prosthetic group to enzymes, or sometimes cardiolipin present in mitochondria. Then fatty acid plus glycerol plus carbohydrate, if it is present, it is called as glycolipid. Example is cerebrosides and gangliosides. If lipid is present in combination with fatty acid plus glycerol, it is lipoproteins. So they are HDL, LDL and VLDL, that is high density lipoprotein, low density lipoprotein and very low density lipoprotein. So importance of lipoproteins, they are present in cell membrane, which maintains the structure of cell membrane. Then they help in a transport of lipids to tissues, and they maintain the structure of mitochondria, microsomes. Then if the combination of fatty acid plus glycerol plus sulfuric acid is present, it is called as sulfolipids. Then fatty acids, they are found in hydrolysis of lipids. So they can be saturated fatty acid, unsaturated fatty acid, or essential fatty acid, or steroids. So saturated fatty acids, it has the hydrogen atoms attached to each carbon atom with a single bond. They are solid at room temperature. So examples are acetic acid, butyric acid, palmitic acid, stearic acid. Unsaturated fatty acid, the hydrogen atom missing, it contains double bond. They are oils at room temperature. So oleic acid, linoleic acid, linoleic acid, arachidonic acid. Essential fatty acid are polyunsaturated fatty acid which are not synthesized in body, have to be provided in the diet. So they are linoleic acid, linoleic acid and arachidonic acid. Steroids, they are nucleus of 3 to 6 membered carbon rings and 1 to 5 membered carbon ring. So importance of steroids, they are naturally present, that is male sex hormones, testosterone, or female sex hormones, estrogen and progesterone, or they can be present in corticosteroids like dexamethasone. Then importance, 1 gram fat gives 9 kilocalorie of the energy. They act as a insulators and protect organs or body. So they are present around the organs and they help the protection of organs, or they are present in fatty tissue, and because of that they act as a shock absorber for our body. And then they store the fats, that is it is a form of energy. And then they are important component of the cell membrane. They form hormones, steroids. They are component of nervous tissue, mitochondria, or the vitamin A, D, E, K. They are fat soluble. The essential fatty acids are polyunsaturated fatty acid, and they have to be provided in diet. That is linoleic, and arachidonic acid. So this is the importance of the lipids. Hope you find this session useful for your understanding and for your exams. Thank you.